Hello, my name is Dylan James, and I did my presentation on PCR and its functions in diagnostic medicine. So first, let's look at this. What is PCR really? PCR stands for polymerase chain reaction. It's an in vitro technique used for amplification of a specific region of DNA whose sequence is known or which lies between two regions of DNA that we do know. Before PCR, DNA ventures could only really be amplified by what's known as overexpression in cells, and this is done with very limited yield and was not very accurate. A little bit more history in PCR and how it kind of led up to its prominence today. 1966, Thomas Brock discovers Thermus aquatics, a thermostable bacteria found in the hot springs of Yellowstone National Park. 1983, Carrie Mullis postulated the concept of PCR and eventually won the Nobel Peace Prize for it in 1993 for it. 1985, Saikai publishes the first application of PCR, beta globulin, and in 1985, Cetus Corporation, scientists isolate thermostator Taq polymerase from T. aquatics, which revolutionized PCR and really kind of made it profitable and brought it into the forefront. There are four main reaction components in PCR that we need. First of all, we need the DNA template that we're looking to amplify. Secondly, we need the primers that we're going to use to separate the pieces that we don't need that template that we need amplified to isolate the ones that we do. Third step, we need enzymes to basically chew off the ends of what we don't need so we can isolate the pieces that we do want. And four, we need buffers to make sure that the solution that this is happening in stays within the pH range that we want it to, so the acidity or basicity does not destroy the DNA. So first, we can look at the DNA template. DNA template simply is just the DNA containing the region that we wish to either look at or be sequenced. The size of the target DNA is a little bit disputed, but from the research I could find, you can use the size of the DNA up to be about, or you can separate out about 3 kilobases of DNA that you could sequence at a time. Next thing you need is two primers. Two sets of primers, usually between 16 and 30 nucleotides long, synthetically produced, meaning you create them yourself by hand. And it is complementary to the 3' ends of the target DNA, and they are not complementary to each other. This is very important because if your two primers are complements to each other, then they will simply bind to themselves and not bind to the targeted DNA sequence that you want, and you will horrendously lower your yield, and that is not what we want. A little bit more information on primers. Primers do not contain inverted repeat sequences to avoid the formation of internal structures. What these structures are things like mutations that can occur because inverted repeats in nature are the parts of DNA that really lead to biodiversity happening and mutations, and we don't want that because we want all of our copies of the DNA to be exact copies of the original sequence that we were looking at. 40 to 60% GC content preferred for the better annealing process. The temperature of the melting of the primers can be calculated to determine the base temperature for annealing to happen, so you know what temperature you need for the annealing to start, and this helps you provide the steps better. Then the last bit is the equation to where you can calculate the temperature of melting of your primer. Third step, enzymes. Usually Taq polymerase or any one of the usually Taq polymerase or any of the natural or recombinant thermostable polymerases. It's stable temperature and it has a melting point of up to about 95 degrees C. It's a high processivity, and Taq polymerase is a 5 to 3 exa only and no proofreading. PCR cycle of operations generally comprised of 3 steps: denaturization, annealing, and primer extension. So annealing, denaturization, primer extension. What you can look at here, which is interesting about PCR in itself, because each time a cycle goes through, where it goes through that cycle, it doubles your copy. So if you start with 1, then you go through one cycle, then it's doubled, and then you go through another cycle, it doubles again because you have 2 things. So it's an exponential process. So you can see here from this interesting chart that, then as the number of cycles, it doubles each time. So by the time you go through 30 cycles, you have over 1 billion pairs. Here's PCR in action within a lab. Because because PCR is so simple and it's easy to follow order of operations, most labs have machines that can do this stuff automatically in these cycles and to optimize the yield. So you can kind of see here, because each step needs to happen at a different temperature, you get this very, very predictable graph of like, here's the denature step, then it goes to the annealing step, then it goes to the extension step, then it denatures again, then it anneals, then it extends. So it's very, very easy to just create a machine that just keeps the temperature at that level, and just each time it goes back to that denature step, it just marks down one cycle, and every single start of a new cycle, you know you've doubled your previous amount. So it's very easy to keep track and very, very helpful, and way to just set it and forget it and come back the next day and have all the DNA you could possibly want. Alternative methods to PCR is something that is used a lot in the forefront today of diagnostic medicine, is RT-PCR, or reverse transcriptase PCR. Uses RNA as the initial template as opposed to DNA, and uses a different polymerase, and it yields the complement DNA strand as opposed to just an exact copy of the DNA strand that you wanted. Steps of RT-PCR is pretty much the same denaturing, annealing, and extension of it, but the main difference is that, like I said before, it creates the complement DNA strand, which is what we want to look for. The practical application of RT-PCR, firstly, it's the predominant method of PCR used in diagnostic medicine. Currently, the main panel used in the CDC test for 2019 nCoV, or more commonly known as COVID-19. It's very useful when looking for the presence of specific viruses, because it allows you to create primers for the RNA, which we could know of, as opposed to and then find the known sequence of DNA and then match that with whatever's in the patient. So if that complement sequence of DNA exists in the patient, then we know that they have the presence of that RNA in them, and they're positive for the virus. So how do we actually interpret the results of PCR? Well, there's a few ways. Most common ways are gel electrophoresis and Southern blot, but however, you could also sequence the entire sequence of the DNA that you amplified. This is usually done when you don't know the sequence that you're amplifying, but you know the primers in the areas around what you wanted to amplify. More applications of PCR. It's used in genome mapping and the function and determination of specific sequences, biodiversity studies or evolutionary studies, to where they put it as if, like, if we go ahead and we have exact copies of the DNA we have now, and we continue doing it for several thousand years, we could look back and have an actual hard record of how our DNA has evolved over this time, and it could give us a lot of insight into how this evolutionary process works. Diagnostics, it's used a lot in nowadays for prenatal testing of unborn children, to test for genetic DNA-based diseases, as well as things like cancer, viral infections. It also can be used to detection of any drug-resistant genes that could cause problems later on down the life, and in criminal justice cases even, there's possible applications for forensic, for DNA fingerprinting, if you could, prove that somebody was at somewhere at a certain time or not at somewhere at a certain time. There are tons of advantages of PCR. The most prominent ones are probably that, one, it's automatable. 2, it is incredibly fast and reliable and repeatable, and those are really the trifecta that any scientist wants to hear when it comes to a piece of equipment and process that they could be doing. It also contains a very, very little chance of contamination, because you can do the entire thing in one room of your lab if you really wanted to. It's very high output because of its exponential scaling and how it copies itself. It's fairly sensitive because you can make pretty much as much of it as you want with no restrictions. It has very broad uses, again, kind of tied into you can use whatever you want for it and test as much as you want, and if you need more, you can just create more. And it is incredibly defined and easy to follow protocols, to the point to where really any person with a high school diploma and the ability to follow instructions could probably use PCR to a decent degree of success and get solid information out of it. So yeah, that was my presentation. Thank you for your time, and I hope you enjoyed it.